Hi everyone, welcome back to glutamate receptors and systems PowerPoint. This video we are going to talk about excitotoxicity and cell death related to glutamate. So I mentioned before that extracellular glutamate can be toxic to cells, can lead to cell death. So we're actually going to talk about specifically how that happens and what that can lead to, or what can lead to an excess of glutamate outside the cell. And then we are going to talk in the last video, I'm going to try to make that the third video today, about the role of glutamate in epilepsy. So let's start with excitotoxicity. So glutamate and other excitatory analogs can be neurotoxic, specifically when extracellular. We talked a little bit about the role of MSG. We have seen that MSG can induce lesions. So we can inject LSG into parts of, say, a rat brain, and it will cause a lesion to that part of the brain. We could also do that with kainate or AMPA or other glutamate or agonists of the glutamate receptor. And what happens is we get an overactivation of the glutamatergic neurons. So because there's an overactivation, then more glutamate is released, and that also will then increase the intracellular calcium to dangerous levels. Because, we talked about how an increase of calcium leads to the binding of those vesicles in the presynaptic cell to then release glutamate. And so that's really what's happening. And then because there's glutamate in the extracellular area, it's actually kind of talking to that presynaptic cell to somehow also then release more glutamate, which then increases the calcium needed to release that glutamate. So there's problems on two fronts. And this can contribute to a different number of pathogenesis of ischemia, ALS, traumatic brain injury, alcoholism, Huntington's disease, multiple sclerosis, and more. This is what's causing a lot of those symptoms of all of these diseases, disorders, is the excess of intracellular calcium and extracellular glutamate. There is one disease that isn't necessarily caused by, say, a lot of these are caused by, say, genetic factors or some kind of injury, so ischemia and traumatic brain injury. And one that's actually due to something you eat is what's called Litigo-Bodig disease. And this is a disease that looks very similar to ALS or Parkinson's, but it's actually caused by eating bat meat that is infected with BMAA. So there's a local seed called Psych seeds, totally saying that right, that contains this beta-methylaminoalanine, or BMAA. So when bats eat this seed, BMAA is then accumulated into the bat's system. And then when the bats are eaten by locals, particularly in Guam is where we first kind of localized this, and that's where these seeds are from, where they're most popular. So we see this most often there. Then people that eat this infected meat will then experience a disease very similar to ALS and Parkinson's. And that's because BMAA is very potent at AMPA, kainate, and NMDA receptors, and acts as this excitotoxin. So it causes an increase in the glutamate release, leading to an increase of calcium intracellularly. Okay, so we've mentioned ALS a few times now with glutamate, and what's happening here is that there's actually a mutation in ALS patients that leads to this increase of calcium, especially in motor neurons, which is why we then see a lot of these symptoms associated with motor function in ALS and Parkinson's and other diseases. And this intracellular calcium, what happens is it actually acts on the mitochondria. So the mitochondria produces this thing called ROSs, reactive oxygen species. It'll normally produce a little bit of this, but like enough that your system can handle. But with an excess of calcium intracellularly, it causes your mitochondria to produce an excess amount of ROSs. So like instead of say 1 part per 100, it's like 20 per 100 or 50 per 100. It's a ridiculous amount. Your body can't handle that much. And that'll cause the cell to pretty much kill itself. So ROSs are very toxic, and this can happen in the postsynaptic neuron. It can also happen in astrocytes. And the ROSs, what they can do is they then go and they can inhibit EAAT2s on the astrocytes, which if it's inhibiting these EAA2s, that's actually inhibiting the reuptake of glutamate and getting that glutamate out of the synapse. So it's actually just increasing the problem. So we now get extra glutamate accumulation in the synapse, and this leads to excitotoxicity in these motor neurons. So really, once it starts, it just gets worse and worse. So here we have a normal functioning condition. So we've got calcium comes in. You can see that here. Calcium can attach to these little guys. A little bit goes into the mitochondria, not a big deal. But here you can see we only have really one of these little vesicles. So we get too much calcium in here that the calcium goes into that mitochondria, and it causes these little purple things or pink things, which represents the ROSs. So that's what can then lead to cell death pretty much. Now, one way we can get to this point that causes these ROSs is via an ischemic stroke. So an ischemic stroke results in loss of blood flow to a certain region of the central nervous system. Okay, so we tend to think of ischemic strokes in the brain. It is technically possible for it to be in the spinal cord, I believe, but usually we see it in the brain. And what this causes is, because there's no blood flow, we also aren't getting oxygen. Okay, so there's a lack of oxygen and then also a lack of glucose, which is needed for energy. So we have a loss of oxygen and a loss of energy. And so that means that energy-dependent processes will fail. So an example of that would be sodium-calcium ATPase. Okay. So we've got our sodium-potassium exchange pumps. Those are going to stop working because we need energy. We need that energy caused by the glucose and oxygen, so ATP and ATPase isn't going to work. So our sodium-potassium exchange pumps are just going to stop working. They're going to freeze. We're not going to be able to get back to our resting potential. This loss of ionic gradient will then cause glutamatergic synapses to just dump the glutamate. What it does is the cell kind of freaks out and says, just get rid of it. It just throws everything out. So all of our glutamate is going to leave the cell. In order for that to happen, calcium is going to enter the cell. So when these pumps fail, when we aren't able to get the gradient we need, pretty much the calcium pumps will just open up, calcium floods into the presynaptic cell, and all the glutamate leaves. Okay. We then see failure of those EAAT transports, and it can actually then reverse glutamate flow. So where glutamate normally flows in, it will flow out. Where it normally flows out, it will flow in, depending on the ion gradient of that cell. So a lack of oxygen causes these major things to happen. So after exocytosis, when all of this happens, it will eventually lead to cell death if it's not reversed quickly. And there's two types of cell death that can happen. There's necrosis, which is an uncontrolled cell death, and then there's apoptosis. Apoptosis is planned cell death. This is like the cell's planning this. All cells are going to die eventually. They all have an expiry date. They're going to apoptosis at some point. Necrosis is unplanned. It's uncontrolled. It happens usually due to some sort of trauma. So, sodium and chloride influx into the cell can cause hypertonicity, which can cause necrosis. Osmosis causes that cell to swell because now there is so much of these ions in the cell that water is going to flood into the cell as well. The cell, it's like a water balloon with too much water in it. Eventually, it's going to rupture. It's going to pop. Okay, so this is what we a lot of times may see during an ischemic, like after an ischemic stroke, is we'll see necrosis, that uncontrolled cell death, because of the inability of these pumps to maintain that gradient. Apoptosis is a planned death, and the cell does this by increasing the calcium. Okay, so it's a planned cell death, planning to increase calcium into the cell. This is planned to then increase the mitochondrial ROS, and it causes that depolarization and swelling of the mitochondria. And when the mitochondria becomes damaged, it then leads to formations of pores in the mitochondrial membrane, which then causes cell death. Okay. So this is also kind of what we were talking about with exocytosis cell death. This is what's happening, but it's not planned. So here we see, here's a normal cell. If we're going through necrosis, we have here, this is what it might look like. Here's apoptosis. So there's those two modes. Both modes are going to be initiated during an ischemic or glutamatergic injury. And in apoptosis, it's planning to die, so a phagocyte will be around to help clean up that debris. But in necrosis, it might not be planned, so the phagocytes might not be around. So now these dead cell membranes and all these things are just floating around waiting to be cleaned up, and that can actually cause further cell death to the cells around it. And that's why with stroke and other brain injuries, it's really important to get to things quickly and try to minimize the injury as quickly as possible so that this cell death that does happen doesn't spread larger. And a lot of times with strokes, that's the thing is, this area of the stroke may be small, but because of this necrosis and apoptosis that's happening, the injury just keeps getting bigger and bigger because the phagocytes aren't around to clean up those dying cells, to clean up the extra ions that are now floating around that can then trigger other cells to also go through this. Okay, so glutamatergic cell death. Both NMDA receptors and AMPA receptors are interesting targets for some sort of neuroprotective agents in ischemia because if we're able to block the NMDA receptors or block the AMPA receptors, then perhaps we could reduce the volume of injury in ischemic stroke because we could then block the other cells around the sick cell, the dead cell, from opening up their channels to then get an influx of those ions intracellularly, which can then cause the cell death. Okay, so we've done these types of studies in animal models, and it does look like antagonists to the NMDA or AMPA receptor does reduce that spread of ischemic injury. But it's been very difficult to translate to humans because when do you give this antagonist? You have to give it, a lot of times in the animal models, we'll give the antagonist before we cause the stroke. And what we see is that by having this antagonist ahead of time, it helps reduce the spread of the stroke afterwards. But you can't just give a human, AMPA or an NMDA receptor antagonist all the time just in case they have a stroke. And then sometimes we give the animals a stroke and immediately give them an antagonist. Again, that's very difficult with humans. A lot of times people don't know they've had a stroke for a while. It could be a few hours, a few days. So it's really hard to translate these findings into humans and then figure out what the best course of action would be in humans. It's also very rare to do clinical trials with stroke remedies just because of the acute nature of the injury. It's usually a one-time thing. Again, it's never planned. It's really hard to identify people that are at risk for a stroke. And, it's also hard because there's more than just ischemic strokes. There's different types of strokes. So when someone has a stroke, treating someone for an ischemic stroke if they don't have an ischemic stroke could actually make their injuries worse. Or treating someone that had an ischemic stroke as a different type of stroke patient could also make these things more challenging. So taking the time to identify the type of stroke also then lengthens and reduces the amount of time you have for treatment. So the whole research into stroke can be very challenging. And I know that actually here at U of A, Dr. Fred Colburn does studies on stroke. I think he does a few different kinds of stroke research. I haven't kept up with their lab's research in a few years now, but very interesting stuff. So overall, glutamate can cause this overexcitation that leads to cell death by either or necrosis, apoptosis, and that's what's called excitotoxicity. So let's stop this video here, and in the last video we'll talk about the role of glutamate in epilepsy.